for covering confocal microscopy. This is a fabulous technique. It can be an innovation and in microscopy history. So I think it's a really important topic. Those who are either developing microscopy or using microscopy, especially for advanced laboratories, many of the biological experiments uses this confocal. Why? Laxman, have you ever used a confocal microscope?
Sorry, Professor, I haven't used confocal microscopy, but in our lab, it's used by our seniors.
Right. So when we use confocal, confocal provides an excellent resolution and also three-dimensional imaging, which is not typically possible with a conventional wide field fluorescence microscope. So this is giving you a great tool for revealing three-dimensional structure of the cells and tissues. So that itself is a big revolution, because previously, just wide field microscope, it's very hard to see three-dimensional structure. But think about the biology and all the molecules, they are all three-dimensional. So that's why confocal is a revolution. So let's get into the detail of confocal. So I first bring a picture what you can do with a confocal microscope. Here's a beautiful image of a retinal cross-section. And this beautiful color means its own like distinguishing cells. There are like photoreceptors in purple, and horizontal cells in the yellow, and bipolar neurons as green. Bipolar means there's a cell body and there's a two waves of extension coming out, and amacrine cells in pink and blue, and ganglion cells. So this cross-sectional image, I mean, I'm not telling you the ophthalmology subject, but here the point is you can distinguish different cells in detail, their shapes. While this one is like kind of collapsed image, the original confocal is in fact like three-dimensional imaging. So this gorgeous and very detailed in subcellular resolution is possible by confocal. So today I'm going to discuss first the history, how we have got to this invention of confocal microscopy, and what are the principles of confocal microscope. And interestingly, confocal has two kinds of imaging mode. Simply, you can have a scattering mode of confocal imaging. The other, you can have a fluorescence imaging with confocal. On the other hand, next Wednesday, we'll discuss about two-photon microscopy. That doesn't provide a scattering or reflection mode. It only provides fluorescence. So confocal microscopes reflection mode is also another very nice ways of giving us a contrast without any label. And confocal, when we say, is usually laser scanning is involved. So we usually call LSM or laser scanning microscopy. And let's think why these are necessary. Also, as I said, confocal provides very nice resolution, which is better than conventional imaging. So that also makes it a choice for people who are looking for more fine detail. And for that, resolution is coming from a so-called pinhole, because the confocal actually needs a pinhole, and that pinhole size in reality or in practice are very much of an issue, whether you use a commercial confocal microscope or those who are developing confocal microscopy by yourself. And we better to compare with the confocal. Why is it somehow great compared to wide field general microscopy, fluorescence microscope? And also, as all these kind of laser scanning microscope, what it means is you are scanning point by point. So what that means is to generate a 2D image, you have to scan point by point. So let's say 1,000 by 1,000 pixels, you have to scan a million pixels at a time. So that intrinsically makes this type of laser scanning very slow, but there are tricks how to make it fast. So with a faster scanning, so we will discuss about the faster scanning method. And also not only fluorescence, if you consider multicolor extension to really a much more of a color scale in wavelength that we call spectral imaging. So I'll introduce spectral imaging using confocal microscopy. So you can turn unmute, I think, as far as I will ask questions with you. But let's first think about the history of confocal microscopy. Here I bring a person called Marvin Minsky, who is at MIT. He is very famous for not for confocal, but AI. In fact, he's a so-called father of artificial intelligence. And but on the other hand, he actually is the first one who invented confocal microscopy while he didn't publish it. How do we know? Instead, he actually filed a patent, which he didn't want to, but some of his relative was a lawyer, and they suggest him to, hey, you've got a better patent. So that's why in history, we actually recognize him as the father of confocal microscopy. But in fact, that year, 1957, in history, the time's important, but somehow you can recognize in Japanese journal, in fact, in domestic Japanese journal, 1940 sometime, 43 or sometime, there's a concept of confocal there. So that's kind of a little bit controversy, but that was in Japanese, people didn't recognize. So right now, I guess the Western power makes the father of the first invention as Marvin Minsky rather than the like unknown Japanese researcher. Okay, that's kind of something interesting in history, but let me put this pattern, which is in 1960 by Marvin Minsky, microscopy apparatus. And this one, we will actually get back to this, but let me tell you a story, how this has been possible. I think that reflects us something to think about. He was a prominent student like you guys, and those who are selected actually got a fellowship. He was at Harvard and he got a fellowship. He wasn't even like majored in this this kind of engineering. I think he was, let me see, I forgot what was his major, but he was interested in neuroscience, in fact. And when he was delving into neuroscience, he realized, hey, due to this scattering of the brain tissue, we cannot image them. So I think we want to solve this problem. So that's how he got involved in this. The important thing here is, I think the power of education and the pursuit of something knowledge and freedom to try something, which is something I think it's very much necessary for you to also realize, because in a given curriculum, you just follow and take exam, and you don't learn much, and you're only following what has been done before. But in his case, he got a fellowship, which means you can pursue whatever problem you want, and all the laboratory in the universe is open to him. And you don't have to have any students, you don't have to have even faculty members, you just pursue whatever you want. I think that makes it really, really good way to doing something, some new exploration. For his case, he wants to solve that problem of solving the scattering issue, and he was looking at the tissue, and is there any way to solve for discriminating this blurry axial resolution. So here's his pattern, two ways. One is called transmission geometry, and the other is a reflection geometry. So I strongly urge you to read the reading material, Memoir on Inventing Confocal Scanning Microscope. This is full of fun, and I strongly encourage you. It's just fun. You have to read, and I think it gives us some insight. So here is this one. This is the confocal microscope he built. In fact, he first time to use his visiting machine shop in the university, try to, using milling machine to make this. So I strongly encourage you, whether you're a computer scientist or you're a molecular biologist, doesn't matter. If you want to do something, you got to learn how to build something, how to grind, how to make a hole. I think this is a really important practice and lesson from him. Okay, let's get back from the history to our confocal microscopy characteristic. So what we are learning is actually the principle was done before. So we have to getting better to understand and ask questions. What are the limitations of confocal? So first of all, we call optical sectioning to reject out of focus background. So all the now imaging process, you make a focus of light and that focus gets back to the image plane of focus. The problem is when you shine the light to focus, the light is not only focusing, but before and after, there are photons. These photons also undergo a reflection or scattering that also go back to your image plane that blurs the background. So how to reject is he uses a way using so-called pinhole, and that names as spatial. Can you guys guess about this? Not that difficult. Reham?
Gating.
Yeah, can you guess what this is?
Temporal gating.
Oh, okay, right. Here's temporal
second,
spatial gating. Yes, it's a good guess. Yes, that's true. While I didn't explain OCT or optical coherence tomography, which we will deal with this later, and that is using temporal gating. What it means is you are making a gate to only the time when you want. That provides, in fact, the axial resolution in OCT case, while confocal uses you only want that focus. So you are using somehow spatially blocking, making only gate. Here, the practical sense of gate means actually pinhole. So we'll get into that. So that provides us somehow improved resolution over conventional microscope. We'll think about it why that's the case. And as I say, application to both. What about non-fluorescent sample? That's possible with confocal, but not possible with multiphoton or two-photon microscopy. But both fluorescent sample, you can apply confocal to both of this. So especially, let's get the first fluorescence, which always you need to have a fluorescent label, or the biological sample. Many times it has also autofluorescence itself. So what if it's not the case? Confocal still can provide some kind of contrast based on light scattering. So that can be really good. And this is called, let me see, Jungmyo?
Reflection.
Okay, great. This why we call reflection, because you send the light and usually the light's reflecting back, we are capturing that. So this reflection mode actually allows confocal microscopy application, not only biology, but also many other important things such as industrial application, or like when you are generating chips and small micro machines, you can actually profile the surface or even biological tissue. So that's why commercialization of confocal in industry level is also very much useful. So let's go to now what makes confocal as be able to take three-dimensional imaging. Let me first show you an example. So what do I mean by three-dimensional imaging? Then wide field conventional ones cannot take 3D. No, you can actually move the focus up and down. You can take pictures. So let's take a look. So these upper ones are wide field, a general one, and lower ones are confocal. So you see the fine, somehow something's blurred in upper side. And even this one, somehow it's blurred. For example, like there's a pollen grain. This is a third column here. And you can put a focus at the middle of pollen grain. Somehow, in reality, pollen grain has different in the shell and inside. But look at this one, wide field doesn't show that detail because up and down fluorescence coming out, it just blurs it. While the lower ones, you can see a beautiful cross section. You are really cut the section and look at, it's like a cut the egg. I mean, baked egg, cut the egg at the middle, you actually see the fine detail. But in reality, you are not cutting with a knife. You are not physically cutting, okay? So that's why we call this, in fact, different ways. So how is this possible? Let's try to invent confocal microscopy, okay? So first of all, you've got to have light source and you send the light. Now you understand what's a dichroic and how fluorescence, epifluorescence microscope work. You need to have dichroic mirror. Look at this, you have focusing with the objective lens. So with this purple line is focusing. Then you have a focal plane over here. And then with this geometry, your fluorescence comes back to the same way. So I put as a green arrow coming back, exactly the same path, and then it'll focus. You just have to put your detector on here. But wait a second. When you shine with the purple line, in fact, in that focal plane, but ups and down, there's a still this excitation light exists. So for example, what happens, the excitation from out of focus, let's say in this more reddish color one, and what happens is this reddish one, you remember from your geometric optics of the lens imaging formula, 1 over S1 plus 1 over S2 equal to 1 over F. That means if the object location is shorter, the image location gets farther. So they move differently. So for here, it's farther here. So your focus will be earlier. And then it will be kind of spreading. So how to block this out of focus light is a question. And the answer is quite obvious in this geometry. Hey, because that focal plane light is making focus here, why don't we put a tiny, tiny pinhole over there? So which allows well for this focal light from the focal plane, but blocks most of the light from the out of focus. That's the key concept. It's very, very simple concept, right? So that's all. So this way you can actually achieve only focal plane information. And you can actually move the focus ups and down to take upper layer, lower layer imaging information. So that we call to reject out of focus light and only permits in focus light. So Soo-hyun, can you guess what that is called?
Optical sectioning.
Great. Yes, because you are sectioning not with a knife, but just optical means. So we name this kind of a XER discrimination by just a light trick of light only. So that's we call optical sectioning. So as I said, let's get into the brain of Marvin Minsky, how he developed his microscopy of confocal. So the first is a layout transmission mode. And this is the simplest one you can think of. Let's say there's a sample one, there's a star sample sending out the light, and that light will focus. This is so-called illumination light. And let's say you have a thick object. That's what makes him bothered to get into thinking obsessed with only this problem. Hey, this P1 is what you want to get the information. And then you have just another, this is for illumination. So something different from wide field imaging is you are now sending a focal light onto the sample. So that's the difference with wide field. And the light coming from this focus P1 will get into another now imaging system. So the simplest one as one lens, and you can have this geometric optics of focus. What about this out of focus light? They will be blurred at this point, so you can put detector just behind this pinhole. So with this one corresponds to this. So focus are common. That's why he named it as confocal. So meaning having the common foci. And here what you can see is a point illumination and point detection. So the information you are getting it is really one point information at a time. But you want this object like full scale. So how can you do it simply? Now you are getting information of this point P1. How can you get all the points in 3D of this stick object? Yeah, the answer is you got to move this point by just simply sample moving like in XY direction for 2D, and you can move in axial direction to obtain this axial or Z information. So that means you have to somehow move the sample. That's called, let's see, Luxman, can you guess?
Mm. Sorry, Professor, I don't
That's
know.
fine. That's fine. So what that means is you somehow scan that point, either moving sample or the point. Okay. However, you have to be practical. This is ideal for conceptually. But let's think about if you have this big of object, the sample. In fact, this geometry is rarely practical because if you have a thick object, this nice geometry may not making sense because of a scattering. So in practice, we rarely use it, but this is conceptually the simplest one. And possible if this object is pretty much very transparent, then it works, all right? So, but as a conceptually, this is the first you can think of a transmission. But like in fluorescence, people normally use epifluorescence. Likewise, confocal also is using that epi mode or reflection mode. So here's the idea, because now you understand how fluorescence microscope works. Let's think about it. So let's have a point source shining onto the sample, so you can make a point object here. And then this lens. And obviously, when you send the light, that light interact with sample at this point, it will be also scattered, back scattered. And that back scattered light, you can collect by putting a beam splitter here. So look at this arrow backs, and then it comes back to the corresponding point of the conjugate plane of point source and this. Here comes, you just put a pinhole aperture over here. What about the out of focusing light? That light will be below and after, so they will focus like earlier or after. They will be all like spread on this conjugate plane. So you just put a pinhole to block those. So conjugate image plane, putting the pinhole aperture is the key concept of confocal, in this case, reflection mode. And that makes the sample like free from the other part. So this is a more practical sense. So that means think about your fluorescence imaging. You shine the light onto the sample and you collect it with the objective lens. Somehow now shining is through the objective lens. The objective lens is serving as the, let me see, Sadaf. Can you guess what this is? Oh yeah, in fact.
Condenser.
Exactly. Yeah. Objective lens is serving as a condenser lens. And here, again, look at this geometry, point illumination, point detection, which requires, like Laxman said, so requires scanning for imaging. Okay, then the question now is, what about fluorescence microscope? So what about fluorescence imaging? Let's look at a little deeper. So here comes this is later method by Confocal and Jeff Richman from Harvard. This is a review paper, very well written. So I'm actually taking their picture for this. So here you can see there is a specimen. You have objective lens and you shine a focus light, and that light will emit photons. And this is that famous 4F system. As the previous Q&A session, why 4F system? 4F system is great in this regard because you can put any kind of filters. At the same time, 4F system, not only 4F, you've learned Fourier optics. Now, here's the place where Fourier optics gets also important because objective lens always have back aperture, and through the back aperture, there is a collimated emission light, and then you have to get this tube lens to focus it down. So this is 1F, 1F, 2F, 2F, like 4F system allows a modern confocal microscope imaging. And what it means is, in fact, that fluorescence is having, so when you shine the light to here, it goes through this optic, so you are making a kind of point spread function of excitation. Out of this, on the detection side, from this light coming in, you are generating emission point spread function. So in the end, the signal you are getting, it is both affected by excitation and emission point spread function. That's the kind of the key when we are generating a confocal fluorescence imaging. So that's one thing you want to understand. What about on the side? Look at this side. There's also fluorescence coming out. There could be, but that is gonna be blocked by the presence of the pinhole. And that this mechanism makes the lateral resolution of confocal microscopy even better than conventional microscopy. Because conventional, think about the conventional, you remove this aperture, you put a CCD camera here, then this and this, they will just get into overlap. Now confocal pinhole effectively blocks the out of focus, not only axially, but laterally as well. Okay, so that's I want to discuss with you. And why do we need a scanning and how we can do scanning? You guys all understand why we need scanning, because one point corresponds to one point and you put a pinhole there. That's the mechanism. So far, we understand. But the important thing is you somehow want to see all kind of this sample in 3D. How can it be possible? The first thing is make the system really simple there. In fact, this is how Marvin Minsky did. And that point of our interest, you move the sample just laterally and axially. So that is as simple. So that we call this way, you build one system, you just move the sample. That we call, let's see, sample scanning. Because you are just simply scan the sample. So this is simple way, but in practice, nobody likes it. Why? Because then you have to put the sample in XYZ translator, you have to move. This is simple and nice, but this is good for proof of principle. But in the real system, for example, like you are doing in vivo imaging, or you have a cells on plate, you don't want to move the cell like kind of XYZ scanning. So that's why this is great, simple, as good as proof of principle, or when you write a paper, this is great. But in practice, that's not the right way. But then you've got a problem here. So how about let's scan the point. So in between somewhere, like there's a parallel infinity collimated light. So why don't we put an XY scanner? Scanner that just means there's a mirror, but it actually can rotate. So it can change the direction of the reflected light. And so for XY 2-dimensional, you just need 2 mirrors orthogonal and that's X scan and Y scan, so that you can move this point, for example, laterally. Okay, that's great. So laser scanning, that sounds good. But now we've got another problem here. So look at that. Let's put a mirror over here, like scan mirror, and you move the point to this position. And from geometric optics, that corresponding point is not here, but here. But the pinhole is blocking over there. How can we solve this problem? And this is a real issue. Yeah, ideally, while you are scanning, you just move the pinhole corresponding way, back and forth. But look at this pinhole is tiny micron scale, and this is totally absurd, and this is impractical. So we've got to solve this problem of laser scanning confocal. All right, so there comes, let's bring the idea back to look at how confocal microscope works. So how about this? Let's send the laser light in. Let's use a beam splitter to send, and let's say here's a scanning system. It goes into the objective and put into specimen. And then instead of putting a pinhole before, you see this scanner changes, right? For scan, the mirror moved, and that position, the light reflects back into. Then what happens? In fact, they come in here, they go back to there the same time, assuming the timing of reflection is way more faster than the galvanometer movement, which is true because the light goes speed of the light, while galvanometer is very heavy object, it doesn't move that much. So this is possible. And that scanning system, in general, we use this way of XY galvanometer. So you look at this laser beam coming in, and this one scan mirror rotates, then makes this X scanning possible. And on orthogonal, another galvanometer also rotates that allows not X, but Y scanning possible. So this is like how we use typically XY scanner. So what do we mean by scanning here? So let me show you one more time. This is such a profound importance to understand. I'm spending time for this. So to and from. So let's say input as a green light. You know, this two line means it's a field, collimate light goes in, scan mirror, and then it goes one focus. And so this is scan lens focus. And then now you start 4F system of tube lens and objective, you focus. Okay, that's fine. But now I want to make a signal from next point over here. How can you do that? You can rotate the scan mirror one kind of several degree. Then it now changes the direction of this red one. So from geometric optics, this red one, this point, that will go that point. Now, think about the reflection light. Reflection light goes back exactly the same geometry of red or green, but that scan mirror position is staying there relatively, so it comes back to the same position. This is the key of this scan mirror, and what that means, the emission light will be not changed after the scan mirror. So this is the point I want to make, and here how the scan mirror works for scanning for this one direction is described it here. And so this ways of scanning, you don't need to change the pinhole location if the pinhole stays after the scanner. Is called, let me see, not. Can you guess how we can call this?
I don't know, Professor.
Okay, yeah. In fact, you know, the problem of scanning is here because it changes the location of the point. Then should we change the pinhole accordingly? That was an issue. But if you look at this scan mirror change, if the detection is going back here, it's all the same. So this means you are removing the effect of scanning, so it's called descanning. Confocal is descanning, allows you don't have to move your pinhole location. And this is assumed by the scanning speed of the mirror is negligible compared to the speed of light. Okay, and so immediate fluorescence can be collected through the objective along the original illumination path to conjugate focal plane at the detector pinhole, so the pinhole doesn't have to change. So that's called a confocal descanning. And what's good about this? This just solves the problem of should we move the pinhole accordingly? No, no need for moving the pinhole and the detector, usually that PMT, which I will describe in Wednesday, photomultiplier tube detector. And this allows a great advantage of confocal, but at the same time, also it has a limitation of confocal. Why? You have to put the detector not right close to the sample, but very far after the scanner. And this one still will be not easy to understand at this point, but in comparison with two-photon microscope, we call that as a non-descan detection. I'm sure this is not making sense to you right now, but after you learn about two-photon, you will understand why non-descan scanner, which means two-photon case doesn't utilize this descanning method. Okay, but that gives, in fact, advantage for two-photon case. All right, so let's go back to the resolution issue of confocal. Initially, I said many biologists love confocal microscopy, or now probably super resolution microscopy, to see even more detail in 3D. What is the shape of the interaction of the molecules and fluorescence and cell organelles and cell-to-cell interaction. And that requires good resolution. So I want to bring an experimental data. In fact, this one looks like a simulation data, and it's XY lateral versus axial. Think about the geometry of imaging. Fundamentally, lateral and axial are kind of different. In fact, axial resolution is usually worse. So that's what you have to remember. Unfortunately, our biology, all the molecules and our world is three-dimensional. However, our imaging is always limited in axial. Look at that, how broad and elongated the point spread function looks like. So one way to describe this is a good way, just normalize the axis in lateral versus axial. So I put it here, lateral unit as a U and axial unit as a V. This is non-dimensionalized units, so that we can compare the relative size of PSF in axial and lateral dimension. So this blue one is lateral, so you can see very narrow, nice PSF, while this axial ones are way more bigger than PSF. So this is the reality, and you have to understand, because we are always, most of the time, limited in axial directional resolution. So I just explained this. This look like a lucky ball shape. Your point spread function, axial direction is always worse. Okay. And what about the quantitative ways? You guys remember in general microscopy, what is the resolution? Remember the Rayleigh criterion, which is coming from the point spread function of a point object. Think about the Fourier optics. You have a finite aperture, which is mathematically defined as a circ function, and the imaging plane is Fourier transform circ is a sinc function. Like that airy disk shape, you can calculate the full width half maximum. In fact, in practice, full width half maximum, but top to the first minimum. That is, this one is so popular, you guys have to remember it. 0.61 lambda over NA. That's your resolution. So just simply, if you don't remember that, it's half of lambda over NA. That would be good enough. However, I can say here, it's not 0.6, but 0.4 lambda over NA. So what that means is, the reason for this is due to the presence of pinhole size. And this one allows confocal microscope about 40% improvement of lateral resolution. So that's why biologists love confocal. While this is so big, expensive, before it was $500,000 apparatus, now it gets down, even down probably 0.2 million or 200K. Still pretty expensive apparatus, but that's what it is. And what's more, confocal provides axial resolution. That is the key aspect. So, but you can see the formula is a little different, 1.4. Here comes a refractive index, and usually axial direction is not only NA, but NA squared. So that's something you guys have to see. How about it in this optical unit of U and V? You can define lateral U and axial V. This is non-dimensional. How can you define? So lateral physical dimensions are, then you've got to have K wave number, which is 2 pi over lambda. So it's non-dimensional, and you have this parameter of NA and V, such as this way. All right. So then, okay, we define this resolution. How does it compare with the wide field microscope in terms of resolution? Here's an example. So this look a little weird. So horizontal direction is lateral and vertical direction is axial. Because the fundamental geometry is axisymmetric, you can just plot in this way to show three-dimensional point spread function. So left one is a wide field, right one is confocal. So wide field has this point spread function, which actually have this part. That's why you don't get a very nice three-dimensional image. It's all blurred out. But confocal removes this out of focus effectively. So that one, but this is not on scale. So I put this data of actually simulation data of the point spread function profile. You see, guess what? The chubby one, thicker ones are wide field resolution, while confocal, you've got to divide by square root 2. So it actually has a 40% improvement in lateral resolution. All right. Also, another aspect is you can see the side lobes over here, but this one is dramatically reducing side lobe. You have so much cleaner image in 3D point spread function. So that allows a nice three-dimensional imaging of confocal microscopy. So why that's the case? So here's the underlying principle. Confocal should make a point for excitation light, and confocal makes another point for the emission PSF. So the resultant point spread function of confocal is really product of PSF for excitation times emission. So if you are using a common objective lens, in fact, this one is square of PSF. That makes it thinner and reducing in terms of confocal. So that's the trick why confocal provides you a better resolution. But here's a practical question. Hey, wait a second. The mechanism of confocal is using a pinhole, and what kind of size of pinhole would be the optimal? So this is all the questions whenever you are using confocal laser scan microscope. So here comes the conceptual way. So pinhole, let's think about pinhole allows you optical sectioning or axial discrimination. So for better axial resolution, you tend to make the pinhole smaller and smaller, right? So better optical section, you want to make the pinhole size smaller and smaller. However, what's the demerit of this? Because your detection signal is behind the pinhole. If you make pinhole small and small, the number of photons going through the pinhole gets smaller and smaller. And we all know that in terms of this detection of photon, the more signal gives you a better signal to noise ratio. So you are having an issue with signal to noise ratio. So for high SNR, you want to get more photon, then you have to increase the pinhole size, bigger. But then what's the problem? Bigger pinhole size means you cannot get a nice three-dimensional optical section. So that's a practical issue. And here's the results of simulation. The detector aperture radius, when it gets bigger, yes, signal is going up, but unfortunately your signal, when aperture gets bigger, the signal to background, the background level gets also a problem. So there exists some kind of optimal radius. So what are the rule of thumb? Is about 50 to 80% of diameter of diffraction limited spot can be used. So let me ask you a question. So you guys now understand the first one. Let me see. 정명? So better sectioning, weaker signal, is it gonna be?
Smaller pinhole.
Exactly. And the opposite, you can say larger pinhole. Okay, that's the opposing, it's all like kind of trade-off, right? And so how does the confocal microscope look like? Here comes the real shape, the commercial one, because you don't have usually the right to open up the commercial microscope. Here I actually show you instead. So the cool thing is they make a good job. So existing nice modern microscope you can plug in, right? So how you can see there's the scan head housing, and there's a dichroic mirror over here. So you've got to need an excitation laser light. So usually here at one point, so in fact, this one was actually used for inventing two photon microscope because they share the laser scanning commonly. Okay, so laser input beam here and neural density filter to adjust the power, and you got to need the dichroic mirror here, and then send the light to go into the XY galvanometer, which scans, okay, the beam. It steers the beam and send into the microscope. But at the same time, remember, you've got to have the emission light, exactly the same path. Now, after this scanner, it descans, which means it exactly followed the same ways of the path. And now you have to make, now this is now descanned position. You have to put the pinhole the right position of the focus, which is confocal position. And after that, you can put anywhere your detector to get the signal. And here's the, like this one, you put it into either on the back side here or somewhere out of nearby the microscope, it's connected. So that's, but also you need a laser system. Some practical issue of confocal is if you have to take a multiple color imaging, you do need a multiple lasers. So that makes this system a little more expensive because as you have like 1 color, 2 color, 3, 4, 5, even 6 color imaging you want, you need 6 lasers. All right, so what are the contrast mechanisms in confocal? We've already mostly discussed fluorescence and scattering, which I said reflection mode. And in fluorescence, 2 types. One is when you are dealing with biological sample, they inevitably contain some autofluorescence, which is sometimes we utilize, sometimes we don't like it as a background, and because it's coming from endogenous. So if the sample contains chlorophyll, collagen, elastin, fibrillin, lipofuscin, NADH, and tryptophan, which is one kind of amino acid. And this one provides autofluorescence. And I would say a longer wavelength, let's say red color, far red, usually have very minimal autofluorescence. While typically what you are more using is a fluorophore of exogenous. If you are using a biology experiment, then you can use fluorescent dyes or proteins, which is like GFP or XFP. Or as I will introduce you next time, quantum dots for targeting specific molecule. So that provides an exquisite contrast mechanism for specific sample molecule that gives a fluorescence mode. On the other hand, from the geometry of invention of confocal, you don't necessarily need the fluorescence. You can just shine the light at the focus, and the scattered light, you can of course also image them as a focus through the pinhole. You can create three-dimensional map of the sample in terms of a scattering mode. So elastic light scattering, good thing is you don't need any labor, right? That's why we call reflection or reflectance mode of confocal. So when you are using confocal microscopy, you can actually use reflection mode, and which do not require any specific filters and the others. You just shine the light and you get back the light. Okay. So, but there are other kind of inelastic light scattering. For example, Raman scattering. I think I'll deal with this in next week, next time. So now let's getting into a little more practical issue. As I said, confocal is laser scanning microscope. It's inherently point by point scanning, or 2D case we call like this. This is called raster scanning. Okay, it's a conventional way of previous TV broadcasting, and at one focal plane. And you have to also make that image like ups and downs, okay, to create a 3D volume imaging. And how can you get there? You know that confocal provides an optical section. Let's say this is a pollen grain. You want to have a cut image, cut, cut, cut. So that resultant image is here. And then as a user, what you want to know is it's a three-dimensional shape. So you can create or reconstruct this image by rendering. So confocal microscope doesn't give you image right away. You have to reconstruct, because it's even 2D, you have to have raster scanning point by point. You just collect that information and you somehow have to reconstruct a 2D or 3D. So now I want to discuss about scanning confocal versus wide field. So always a good idea to compare what you know before and what you are like getting a newly. Let's compare. So this is wide field. You guys all know already. You shine the illumination light through the dichroic. You shine the specimen in a wide field. And that emission light from all over the specimen, you can collect it. So what would be typical detector? Laxman, you're using fluorescence microscope. What will be your detector over here?
That detect the fluorescence.
What do you use in the laboratory? It has to be white.
Yeah.
Have you ever seen what's there?
I have seen. I forgot, Professor, but I have seen that.
Yes. No,
yeah.
it's simple. You are using camera over there, right? Right? So that's camera. And here I put CCD camera, or charge coupled device, which actually got Nobel Prize for this invention. But now in science, we are using more CMOS camera, which gets more advanced nowadays. Okay, so you are putting camera over here, all right? But confocal, as you remember, it's a point by point scanning or information only from a point. So you don't need a camera, in fact. You just need one single point detector in confocal microscope, right? Because you are through the pinhole, all the information you are collecting is supposed to come from one point information. If it's camera, it's a 1 pixel camera. And so you've got to use a very high sensitive and fast and good 1 pixel camera. In our case, typically we are using PMT, or photo multiplier. In fact, its shape is a tube, so P photo multiplier tube. So these are a kind of difference. And I put this a busy slide for your own reference so that you can actually later to study this, read what are the differences between what you are familiar with the wide field versus this confocal laser scanning microscope. All right, so that scanning mechanism, I introduce you only the simplest one, a simple galvanometer which rotates in X and Y direction. Okay, that's called linear galvanometer. Okay, that's most of the time your laboratory of confocal microscope has. But you have to think about how that actually works to sweep all the two-dimensional imaging. And that requires a we call raster scanning. So let's think about that. But unfortunately, this one's inherently slow. So if you have a knowledge in, let's say, in physics, you know that in the concept of inertia, the mass to rotate moving is hard. So one way is very fast movement can happen when this mirror has at its resonance mode, which means at its resonance, it can move very fast. So one of these X and Y. So when we move, make an image, usually we move X and one layer down and Y1, XY2, like this way. So think about X requires a faster scanning compared to Y because Y kind of stays once, have to wait until you have the 1X. So Y scanning doesn't require fast. So you can put Y scanner a big mirror because you don't need a very fast. You make the X scanner a little smaller and smaller and smaller. You want to make it resonant. Then this can be very, very fast. So this is a way of trick to make a resonant galvanometer because you can oscillate a mirror at a sinusoidal instead of a kind of sawtooth wave at its resonant frequency. Typically, we use a 8 kilohertz or 12 or even 16 kilohertz, only in the X direction, because that's the place where we need a faster scanning. And this makes us a video rate scanning. So video rate imaging is possible if you are utilizing resonant galvanometer. In fact, our microscope actually have this resonance galvanometer that allows 30 hertz or even 60 hertz imaging possible. There are other ways of trick to make this X scanning better. One way is you make a cylinder and then you attach a tiny mirror on side, and instead of like mirror moving X and Y, just rotate the cylinder while sending the light laser in. And when the mirror, okay, this way, when the mirror rotates, the reflection laser will sweep this way. But you make this a polygonal cylinder and you put a linear mirror, tiny mirror, and you just sweep it, rotate fast. So that we call polygonal scanning mirror. And this also allows a very fast scanning. And I want to show you in picture. So linear galvanometer makes it this XYZ scanning, and that's coming with this scanner X and Y. But resonant galvano replaces this X1 with tiny mirror because you've got to move lower mass makes your resonance frequency higher, and you can make it sinusoidal at resonance. Polygonal mirror makes this mirror with a rotating direction, and all these facets are small tiny mirrors. By rotating this fast, the input laser is staying still, but output is, by changing this angle, output is sweeping. So that's how we can scan very, very fast. This is a trick. There are other mechanisms which I will discuss later on. But also there's another invention of this super fast imaging called spinning disk configuration. And also another, actually our laboratory developed also this new ways, which is slit scanning confocal, which makes, instead of point, you can create a line, and then you scan this line in one direction only. That covers a whole image. So it's going to be order of magnitude faster than the previous, okay? And another way is, hey, wait a second, all of this technique requires a mechanical movement, which inherently limits the speed of scanning. So people have been thinking, is there any way no use of mechanical movement? Yeah, there exists. One way is you've learned from free optics or grading. We can have this a crystal and sending a ultrasonic sound waves to make a standing wave, then you can create an arbitrary grading over here. Remember, the grading has a diffraction angle, and first order depends on the period of grading. So if you can change the sound wave frequency, this diffracted light first order will change. This is the fastest way of creating a scanning, and there's no mechanical movement, so you can make it even faster, like megahertz range. That's possible. That's just amazing. However, the alignment and diffraction, there are tricks which makes it difficult, but this is a modern technique. You can make an extreme high imaging, such as like kilohertz scanning. You got to use this one. All right, so in especially confocal, there have been interesting development from Japanese team, so-called a NIPCO disk, and that concept is here. So the point of confocal microscopy, one single point at a time, that limits. How about making multiple points and multiple detection as a confocal unit? That's the concept. So instead of one point, there are multiple points simultaneously getting, let's say here, the white dots are actually confocal points. And then when you rotate the disk, the next time you will have all this red one, which is the missing part of your image. So this way, disk is very easy to rotate, so you can collect data very much fast. So light, but the problem over here is you are not collecting the light out of this pinhole, so you are wasting it. So Yokogawa realized the problem of this NIPCO disk, and can we put a micro lens so that you don't waste the excitation light by using this? So that this is a great idea. So we now use this one. So here's a collimated laser illumination, and you've got lens disk array, and that gives a this a pinhole array and illumination light through the pinhole confocal. And now instead of one point, you've got multiple points of emission. And then by rotating it, you get to this confocal image. And this is a great way. It's a way of parallelizing your points to detect separate from each other. So now you don't have to use a single pixel detector, but you can use a fast CCD camera. So that's a revolution, and I just want to show how it looks. So instead of laser scanning, this spinning disk has a really little different shape of emission, and one shot looks like this, but every point after shot, it will move so that you synchronize when disk speed is 0 is there, but when you rotate this point will cover the all two-dimensional area to create a real image. So that's a scanning pattern in spinning disk confocal. And lastly, I want to describe, hey, wait a second. There are another dimension of contrast or information if the sample has its distinct spectrum. But spectrum can only be measured by using prism or spectrometer. How can you do that? Okay, confocal is a one-point detection. Why don't we point the light through the point? We send it to the prism and then measure the spectrum of each point. Then you are adding one more dimension of not only X, Y, Z, but lambda dimension. So it's a multi-dimensional, we call spectral imaging. So even one pixel you can create, if you measure different wavelength of the amount, then we call lambda stack, then even single point, you can create this nice spectrum. So this is spectral imaging. If this is a lot, we call hyperspectral imaging. So that allows us another ways of discriminating contrast to the sample, it's point by point. So that's another amazing rich information from the sample you can get. How can you do that? You need to put that emission light, you need to have somehow a prism. Simply here, instead of prism, you can do diffraction grating, and you can have a slit over there. And yeah, here you can have a real prism to separate the color. And also diffraction grating, you can move the slit one by one through different colors. That's one way, but now even better ways, you can have like all these detectors with different color. You can create the spectrum right away. But this one's more expensive because you need a multiple PMTs here, which called multi-anode PMT. So it's more modern technique, but this is definitely very much interesting, giving another level of information. So here, now I want to finish. Today is a 1 hour lecture, but we've discussed about how we got to invent this concept as a civilization. It was 1957, about 60, 70 years ago. It's not that long time ago. And you can even develop this one, right? And we discussed about the principle and how we can make this one from transmission and reflection mode. And but it's a point concept we somehow need to scan with the laser scanning. And we realize the resolution of confocal is better. And for that, you probably need a proper optimization of pinhole size. And confocal is different from wide field in terms of it provides an axial resolution by optical sectioning, and even laterally, it provides 40% enhanced lateral resolution. However, this inherent point-by-point scanning allows confocal to be slow. So we developed how to fast scan the confocal by making resonance galvano or polygonal mirror or even NIPCO disc. And furthermore, you can put a prism for the emission light so that you can collect a spectral information. And next time, we will discuss a multiphoton laser scanning, and that has a pros and cons compared with confocal. So I wish this time you have get to a chance to know the a nutshell of confocal microscopy. You can write down your questions. I'll give you 2 minutes through the Basecamp. Okay, thank you for your attention, and I'll see you guys next time.